Hello everyone, and thank you for taking the time to check out my poster. My research focuses on the effects of lactic acid bacillus casei and milk on intestinal health. Here, I am focused on their effects in a mouse model of inflammatory bowel disease. The mouse model presented here utilizes dextrin sodium sulfate, or DSS, to induce colitis. Before inducing colitis, mice were fed a Western diet for 7 days to mimic the high-fat and high-sugar diet commonly consumed in the United States. A week after starting the mice on a Western diet, mice were fed milk or PBS with or without probiotic LKCI strain BL23 for 4 days, and then given DSS in their drinking water for 5 days. After removing DSS, mice were then allowed to recover for a week. It is important to clarify my treatment groups before moving forward. The first is my healthy control group, or the sham group, that consists of mice fed PBS without LKCI and no DSS in their drinking water. The remaining mice were given DSS in their drinking water and consisted of daily administration of PBS or milk only, or PBS or milk with LKCI. At the end of the study, the colon is then excised and split for both histological and electrophysiological analysis. We observed that there were sex-dependent responses to DSS, milk, and LKCI. It is expected that mice will experience significant weight loss during DSS administration that will continue a few days after DSS is removed, and we actually see that for both the female and male mice. Female mice experience greater weight loss, which actually led to some being euthanized before the study's endpoint, which are counted here. While both male and female mice given milk exhibited improved weight recovery, which are the filled in squares, black squares, only the female mice given BL23 in milk share similar weight recovery, which are those in the not filled squares. Female mice also exhibited greater colitis severity as determined by histological analysis, right here. But the severity was actually less in mice fed milk and milk containing LKCI, which are these two groups here. Interestingly, all male mice showed recovery from DSS-induced colitis based on histological analysis. But the male mice that were given DSS still had significantly reduced colon length. Among the females, we actually see that there's only a significant decrease in the colon length with just the PBS only group. We also observed DSS-induced disruption to the total active ion transport across the colonic epithelium, which we see in both the female and the male mice. Looking at the male mice that were given LKCI and milk, we see a recovery in the total active ion transport across the colon. And we see a similar trend followed by milk. For the female group, we see a similar trend, although not significant. These findings indicate that milk may support recovery from episodes of intestinal inflammation. Variations in the effects of LKCI-BL23 between delivery matrices and mouse sex support ongoing efforts focused on understanding how the food delivery matrix affects probiotic function in the gastrointestinal tract.